So we were talking about these protocells, and the interesting thing about them, again, with what you've been learning through this class, is this biphospholipid layer, and this layer we don't know. Yeah, it is biphospholipid. Remember, it's going to create this boundary between the water and itself. So you have very similar to our cell. So you have this particular cell protocell being created and has been replicated in the lab, where you have silica inside, you have polypeptides, you have protein type molecules in the biphospholipid layer, and then this particular cell has the ability to exhibit some reproductive properties. Think of binary fission. It can actually pinch apart and turn into two, and also has the ability to do some metabolic activity in the fact that it can incorporate things from its environment into itself. So it can do endocytosis, exocytosis. So it's starting to exhibit some of the behaviors that we definitely have associated with life, with biology as we've studied it. Now, they did find that the increase, that adding clay increased the rate of vesicle formation, and one of the reasons why they think that is clay creates clay is laid down in layers, and this is microscopically. If you look at clay particles in soil, it's laid down in layers, and those layers give a place for these protocells to adhere. So adherence to something, instead of just floating around in water, but this ability to adhere to something tends to be an important property with regard to the creation of photocells. So we started out with compounds into photocells, and then the next thing we need is the ability to pass on our inherited data. We've talked quite a bit about DNA and the ability to pass on inherited information, but the first genetic material was probably RNA, was probably that ribosomal nucleic acid, not DNA. Ribosomes have been found to catalyze many different reactions and can make complementary copies of short stretches of RNA. So RNA, which tends to be a simpler molecule, tends to be shorter, has been shown to have the ability to be able to replicate itself. So it is thought that in the early time, it was an RNA world, not a DNA world, that most of these protocells, vesicles, had RNA in them also that were capable of replication. The thing about the DNA strand is it tends to be more stable. So the fact that you have that double helix, and the double helix with the nucleotides that only bond to the appropriate nucleotide, so few nucleotides, just those four, and then that stability of the double helix makes the DNA more likely to be able to create much larger organisms, much larger species. So the fact that we started out with RNA, but then evolved into a molecule more like DNA, allowed us to have larger and larger life. One of the main ways that we study all this information is via fossils, which most people are probably very, very familiar with. Fossils are excellent and certainly have gotten us very excited about things that happen before we visually could see, and certainly have taught us a tremendous amount about mass extinctions. But fossils do have their limitations, but they also have taught us an incredible amount. The fossil records reveal changes in the history of life Earth. Like I said with our first slide, the fact that there were whales in the desert in some billion years ago And the type of rocks we mostly look at are sedimentary rocks, mostly because the rock is deposited in layers, which we call strata. And those layers, which we call strata, as long as we can prove they haven't been shifted by earthquakes or volcanic action, etc., can be dated just based on that one's on top of that one. And so dated by the most recent fossils are at the surface of the strata, and the oldest fossils are at the bottom. Again, as long as it hasn't been shifted. We have found fossils in sedimentary rock throughout the entire world. And what you have in this particular image are fossils that have been found in the various different strata, going from 100 years ago down to 3,500 years ago. And particularly these, and we'll talk a little bit about these stromatolites, which are very interesting, and they have found, they are the oldest fossils they found, these stromatolites. They found these in Australia. They may be other places, but particularly in Australia, highly undisturbed, very, very pristine fossilized stromatolites. Sedimentary strata reveal ages, but the absolute dating is done via isotopes. And so with isotopes in a very, very constant rate, a parent radiation number decays into a daughter number or isotope number at a constant rate. So that rate of decay can give you an idea with regard to a timeline. It can be used for fossils up to 75,000 years old, which we've been talking about fossils that are much older than that. So radiocarbon dating is useful, so isotopic dating is useful. Sedimentary rock layer dating is useful, but both have limitations. One of the other things, just it's not in your textbook before I move on to this, we have dating with regard to looking at crystals in rocks, and there's also ice cores. They can go into ice cores in terms of glaciers and do some dating based on ice cores also. So there are other ways to date what was going on in the earth at a certain time based on what chemicals were there, what the atmosphere might have been like, and so making hypotheses as to what life forms might have lived at that time. So there are some more cutting edge type of ways to date date the past, I guess I might say, which sounds like kind of a funny thing to say. But the first mammals, mammal type species was a pteropod. And so these were some of the first vertebrates walking that were terrestrial. And most of the reptiles, the dinosaurs, birds, mammals all evolved from the early pteropods. One of the things that we have found is this gradual change or evolution of certain things that we consider very mammalian, so very unique to our particular group, whether it be this hinged jaw, whether it be the reproductive cycle similar, whether it be walking on all fours, or bipedal, or all fours, or even being on land. What you are looking at, there are fossil records that can be traced to look at that development, certainly with some holes of clearly, because again, and I have it on a later slide, fossils have a caveat in the fact that in order for somebody to be fossilized, a species, they had to have something that was hard, like an exoskeleton or a skeleton. They had to have been abundant, and they had to have died in an area where it's likely that their fossilized remains would be still visible to us, like the sedimentary rock. And then there's the element of chance that we can actually find those fossils. So what you have in this image is from 100 million years ago down to 195 million years ago, the changes, the subtle changes we see in this hinged jaw that makes this hinged jaw in the late 195 million years ago, makes this hinged jaw into something that is closer to what our jaw looks like, or a jaw of a bear, or a jaw of any mammal that has a hinged jaw. And fossils are incredibly valuable for that kind of information. Other events in terms of, remember, this is an overview of what happened in the development of the Earth. There are many events that led to the capacity for great changes that we've seen. Some of those events are mass extinctions, of course, and some of the events are just the gradual change maybe of the atmosphere, or change of even something that maybe seemingly you don't think about, but that the oceans became less deep. That you had areas of the oceans, it might be volcanic action, where the land mass came up in the ocean, so you had shallow waters then. So you have many of these things happening as the Earth is forming, and when they do form, whether it's a mass extinction or some sort of geological change, you have species moving into that. You have them adapting and moving into those environments. We divide geological records into eons. Those eons are so grouped. It may be by more likely at this point, probably the best way to group things is by extinctions. So you have a new eon starting when a mass extinction, for example, between the Permian and the Jurassic, you had a mass extinction at the end of the Permian era that allowed for the development of the atmosphere and the parameters for the dinosaurs to reign. And then you had, at the end of the Cretaceous era, you had another mass extinction of the dinosaurs, and we've heard a lot about that particular mass extinction, whether it was a meteor, whether it was a volcano erupting, what particular event caused the mass extinction of the dinosaurs. But that allowed for the rise of mammals. So a lot of times with eras, we divide them up based on major things that have happened. Then another timeline, many times when the Earth's history is being talked about, you look at a clock, because it's something we can relate to. And by the way, if you look at the entire clock and it's 24 hours, we're only about 4 seconds of it. So of the 24-hour clock, humans, and this is, we're talking about the whole humanoid evolution, is only about 4 seconds of it. And as you go through this clock, most of the time is spent in the formation of the Earth, and then the development of the atmosphere that is more conducive to cellular respiration than any other portion of the clock. So the actual, what we have found in terms of organic life, or I guess you might say life that we would recognize, is maybe just the last hour. And remember, we're just the last 4 seconds. We talked about the stromatolites already, and it's kind of exciting how they have looked at this. And these stromatolites can still be found. They're still in existence, particularly in places in Australia, where they have stromatolites that they can actually study that have, we don't know that they've been here 3.5 billion years. We assume they probably have not, but they have been in this particular in a very similar form for all those years, so they can be studied. And they were some of the first photosynthesizing organisms. We'll pick this up on the next one.